when Linnaeus first got started, he had things that moved, Animalia, and things that didn't move, i.e. Plantae. Now, at the time, people did not understand the difference, at least in the Western world, between plants and fungi. But then, people started using microscopes. So there's a bit of a problem with this. Most bacteria have cell walls. Some other single-celled organisms also have cell walls. Okay, they already knew from looking at plants that plant cells had cell walls. Cellulae actually came from originally a term meaning small room. So it was looking at a piece of wood that the cell theory was established. So if these organisms, a lot of them have cell walls, there's also the problem that they move around. By the way, animal cells don't have cell walls. So yeah, that's a bit of a problem, isn't it? So they decided, hey, let's use separate categories. Now, what they also recognized was the fact that some of these cells, when stained with certain pigments, happen to show a big dark nucleus, or a big stained nucleus. And other cells, usually smaller, didn't. They also noticed that these cells with the nuclei also had little bubbles and stuff, other complexities inside. So they call these eukaryotes. Karyon as in yolk, and eu as in true. And prokaryotes are before yolk, because they concluded that the more complex organisms must be higher in the hierarchy of life that they believed in at the time than the organisms that did not have a nucleus. Technically speaking, evolutionarily, this is not true because they should all have had the same amount of time to evolve since divergence. But they believed it as such at the time, and in the end it did turn out that prokaryotes came before eukaryotes in terms of when they first arose. So, people started using terms such as monera or bacteria for the single-celled organisms that didn't seem to look like plant or animal cells in terms of internal structure. And then they called the ones that did, the eukaryotes, this is small eukaryotes, protists, or kingdom Protista. Now this is actually a defunct term because progress has been made to the three-domain system, and protists are not a monophyletic clade, or in our current terms, not a single clade. They don't include all the descendants of a single common ancestor because, well, there are protists that are closer related to animals, closer related to fungi, or closer related to plants than they are to other protists. So if we wanted to say protists, well, we basically included all of eukarya, the domain. But it's still used as a term because it's a little bit too convenient to let go of, to say, oh, small and or unicellular eukaryote that isn't a plant, an animal, or a fungus. It's just easy to use, hence why it's still used. Now these, when they first decided to split out fungi and protists, and then bacteria, or monera as it began, that was later split into bacteria, or eubacteria, and archaea, or archaebacteria, as you may see in some really old texts. That's because it was found that these had certain biochemical differences that made them very much separate from one another. Now this is the old 6-kingdom system. So as we can see, we have fungi, we have Protista, we have plants or Plantae, and animals or Animalia. And we have these two. Now we use a different system in the sense of, hey, Since some of these protists are closer related to plants than they are to other protists, they are not a monophyletic clade, we should lump all of these eukaryotes into a single domain of eukarya. However, the old Six Kingdom system is sometimes still referred to, just because protists are such a convenient catch-all term. So here's some characteristics of the old Six Kingdoms, which for the most part still apply to some degree, other than Protista. Eubacteria is only a few tens of thousands at most of described species to date. This number is a little bit old. They are prokaryotes. They have cell walls. The cell walls contain a unique compound called peptidoglycan. Archaea generally don't. Now, cells are variable in shape and size. So they could be round, they could be rod-like, they could be comma-like, or a variety of different shapes, such as a corkscrew in shape. Now, in other characteristics, they have diverse means of obtaining energy and nutrients. So this could be photosynthesis, this could be chemoautotrophic, i.e. it gets its carbon from reduced carbon sources, but it gets its energy from, say, sunlight. Or it could be heterotrophic. Now, heterotrophs are literally other eating, so they have to get their carbon and their energy from elsewhere. Now, all of them can reproduce asexually, but they can also reproduce sexually in a sense because they can undergo conjugation, where one cell extends a pilus and joins with another cell to allow DNA to flow in a particular direction. Now, it could be that they export a plasmid, which usually manages to make its way across. A plasmid is a small ring of DNA, or ring-like structure, that some bacteria possess in varying numbers. Or they could try to send across a copy of their whole genome. Now, this usually doesn't work because the conjugation doesn't last long enough, so it breaks. That piece of DNA that's now inserted into the other cell, it may undergo some crossover with the cell's local DNA, and it eventually gets degraded because it's not a whole ring. But that still results in, well, genetic recombination. We have traded genome pieces, or at least traded it, given it one way. Now, examples are, say, Escherichia coli, or E. coli, one of the most common denizens of our large intestines. There's Salmonella, there's nitrogen-fixing bacteria. There's actually quite a number of groups in eubacteria, which will be covered in more detail later. Now, for Archaea, we have this is a bit of an old number, so now we have a rather greater number of described species in this, but you notice that it's much less than eubacteria. Why? Because for eubacteria, well, they have fewer extremophiles compared to Archaea. As in, most Archaeans that we're familiar with tend to be extremophiles, although there are a few exceptions. And what does that mean? Well, they come from extreme environments. Are they going to be easy to culture in the lab compared to eubacteria, which often gets found in less extreme environments in much greater abundance than the non-extremophile Archaeans? Well, okay, it's pretty obvious they're not going to be as easy to culture in the lab. Therefore, fewer species will end up identified, because traditionally you can only identify a bacterial species or Archaean species if you can culture it in the lab. This is, of course, a problem. So Archaea are also prokaryotic. Their cell walls and membranes have a unique structure that is very different from bacteria. That's how they were identified in the first place. And most of them are fairly small. Some bacteria can be so large they are easily visible to the naked eye. This does not hold well for Archaeans. Now, some of them colonize extreme environments, others live in the digestive tracts of various organisms, not necessarily just mammals, and they also live in marine environments. Now, all of them can reproduce asexually, i.e. just by dividing, after copying their DNA, of course, and some growth, because otherwise, cells that start off too small are at a disadvantage. So they include extreme thermophiles, which live at high temperatures. They can include methanogens, which generate methane. This, say, in swamps. And they can include halophiles, which live in very salty environments, and so on. Now, Protista have a pretty large number of described species because they tend to get identified from, say, fresh water, and they're relatively easy to culture compared to bacteria and archaea, and or relatively easy to identify under the microscope. Now, they have extreme diversity of cellular structure. This kind of explains to us why they're relatively easy to identify under the microscope, because bacteria, oh hey, rod-like bacteria. How can I tell the difference between these many different rod-like bacteria? We don't have any internal structures to go off on. Well, at least none that can be easily seen with early microscopes. So some have chloroplasts, some have cell walls, some have both. Others have various other complex structures such as apicomplexans. Now, they may be heterotrophic, they might be photosynthetic, or they can be both. So they have variable forms of movement. Some are essentially immobile, root themselves in place. Some are not, such as this amoeba, pictured here. Kelps and green algae were considered parts of Protista. This might sound surprising given that kelps do have a resemblance to, say, plants, but brown algae, which forms most of kelps, is in fact not a plant. Now, they reproduce sexually and asexually. They usually live in aquatic or other moist environments because otherwise the cell membranes dry out and disintegrate, and they have a tendency to die as a result. They have variable forms of movement, as mentioned. So what other forms can you think of besides extending part of the cell out and pulling itself forward? Well, there's always the flagellum. The flagellum is very different between eukaryotes and prokaryotes. In prokaryotes, it whips around like a corkscrew and is one long chain of flagellin, which is a protein. In eukaryotes, it's a much more complex structure, and it whips back and forth, side to side, instead of in a circle. Now, what about the next group? Well, fungi. So fungi, we also have a lot of described species, once people finally got around to distinguishing them from plants, at least. Because, well, they have cell walls, which kind of looks like plants under a microscope, if one is not too careful. They have no chloroplasts, though, which is, of course, a major distinction. Although, to be fair, if you look at, say, say you have an onion. You cut it, you peel off the inner really thin layer, you put that in a little drop of water on a microscope slide, and if you look at it, the cells there don't seem to have chloroplasts either, which means that the confusion between fungi and plants, due to their shared cell walls and their shared stationary structure, is perfectly understandable in an age when microscopes were either not invented yet or were very primitive. Most fungi are multicellular. Chytrids or chytrids are an exception. The cell wall is composed of chitin. Chitin is something that we're probably more familiar with when talking about insect carapace, like beetles. Chitin is similar to cellulose, but there's a few differences, namely a nitrogen-based group that is attached to one of the carbons. So this changes the properties of chitin compared to cellulose. And what else? Well, okay, we can talk about chitin a little bit more later. Chitin happens to be used for naturally degrading thread in surgery, but that's another matter. So, fungi are different from plants in very significant part because all of them are heterotrophic, so they are not autotrophic at all. Plants can be autotrophic or heterotrophic. Why? Some plants have evolved to be parasitic. Some plants have evolved to be so parasitic that they can draw upon fungi or other plants for food, which means that those plants happen to be heterotrophs. Now, fungi are all heterotrophic. Most of them are saprotrophic, i.e. they feed on dead organisms. Most of them are terrestrial, and they reproduce sexually and asexually. Please note that water molds and slime molds are protists, not fungi. That's why fungi are mostly terrestrial. Chytrids are, of course, an exception. They infect amphibians and cause lots of deaths each year, but, well, we are probably more familiar with stuff like mushrooms. Mushrooms happen to also be easier to identify than fungi that aren't so obvious, just because, hey, specific mushrooms have very specific shapes, and of course, the ascocarp or basidiocarp, depending on the mode of reproduction, well, they differ greatly in appearance, and this is really obvious to the naked eye. Especially because eating the wrong mushrooms could be poisonous. Thus, people learn to identify different mushrooms really early on, long before any thought of classifying life or formalizing it or anything like that. So then there's yeasts and molds. Now, yeasts can sometimes also contain plasmids, kind of like bacteria. We can even build artificial chromosomes and insert them into yeasts to be replicated if need be. Please note that these molds in group fungi are not the same as slime molds or water molds as exist in protists or Protista, which admittedly is a defunct group. So plants, we have lots of species of described plants. They are eukaryotic, all of them are multicellular. They have cellulose cell walls, they have chloroplasts or chloroplast-derived structures because there are parasitic plants. Now they can be autotrophic and photosynthetic, almost all of them are. However, there are a few that are decomposers or are just parasitic. Some are parasitic in the form of using other plants to get high up so that they can photosynthesize up high without actually having to grow there. Some are parasitic in the form of strangling vines or similar, climbing up other plants. Some are parasitic in just tapping into the vascular systems of other plants and drawing nutrients. Or some just draw water. Most are terrestrial. Some plants have gone back to aquatic ecosystems, but for the most part they tend to grow on land. Now they reproduce sexually and asexually. How do they reproduce asexually? If you've ever seen a strawberry plant and its runners, well, that's asexual reproduction right there. So these can include mosses, ferns, conifers, and flowering plants. This basically, this list here is pretty much a catch-all of the four main categories of plants. The first are nonvascular plants. They don't have specialized vascular tissues to transport water, minerals, and other nutrients, such as photosynthetic products, around the plant. They rely on diffusion. Second, we have seedless vascular plants. So they don't have seeds, but they do have vascular tissue. And then we get into seed plants that are not flowering, so conifers. For now, we can conflate those terms with conifers. And then we have flowering plants, angiosperms. Admittedly, some gymnosperms, i.e. naked seed plants, they do not have needles like conifers do, but conifers are the most obvious example. Some of them, though, are broadleaf, like, say, ginkgo plants. So, animals. We have lots and lots of described species because they are obvious. Compared to, hey, I have this sneaking suspicion that this tree here, which looks almost exactly like that tree over there, is not reproductively compatible. Yeah, no, that's not nearly as relevant to human notice or the Mark I eyeball as, hey, there's an animal that looks like that. Hey, there's an animal that looks like that. These two beetles look very different. Do they reproduce? Well, initially, due to the morphological species concept that was used early on, we could often find different stages of the same organism being misclassified as different organisms before the error was noticed and corrected. Thus, animals, due to the fact that they move around, are really, really obvious to the human eye. Things that don't move have a much greater chance of being overlooked, like, well, basically everything else. Or in the case of bacteria and archaea, are too small to be casually noticed by basically everybody. Now, of course, we do advise noticing plants, especially when walking around in a forest, otherwise it will hurt. So, animals are all eukaryotic, all are multicellular, they move, at least that's why they're called animals. They have no cell walls or chloroplasts. Now, all are heterotrophic. Most will reproduce sexually. There are a few animals, such as bdelloid rotifers, that happen to reproduce asexually. Aphids can reproduce asexually. Now, animals live in terrestrial and aquatic environments, such as, let's think of a few examples. Well, aquatic insects happen to be a very good indicator of water quality because they are immersed in it most of the time. However, many of them still need to breathe air, the ones that don't use gills, at least gills of their own, as opposed to physical gill, which is a bit of a different concept, but that's reserved for aquatic entomologists mostly. So examples of animals that we can think of include, well, obviously elephants. There's also maybe some beetles wandering around in this picture, but we don't see them. There's sponges, corals, insects, snails, birds, and humans. Now we might be thinking, hey, wait a second. Sponges and corals don't move around. Why are they considered animals? Well, say we run a sponge through a strainer of some sort and break up all the cells. The cells will move around and reassemble a new sponge. Corals, well, they are animals because, well, they are part of the Cnidarian group, in the polyp stage, to be specific. Cnidaria includes, oh, by the way, that's spelled like this. Cnidaria includes stuff like jellyfish and hydra, as in not the mythical nine-headed snake or serpent or whatever, or dragon, but a small organism that has a body that's essentially shaped like a blind-end sac with a few tentacles coming out near the oral end. One of the disadvantages of such a structure is that you cannot take in more food before the previous item is digested unless you want to spit the previous one out. So, domains of life. Now, modern phylogeneticists have this thing about, hey, we need each clade or group that we identify to all descend from one common ancestor and to represent all the descendants of that one common ancestor. This obsession may or may not be a good thing, but the domains of life were proposed in 1996 by Charles Wolsey, who did a detailed analysis and revealed that all organisms fit in these three clades. So, he created a new taxonomic level of domain. So we have domain Eubacteria, domain Archaea, and domain Eukarya. Now we notice that Archaea are closer related to eukaryotes than they are to other prokaryotes in Eubacteria. This was basically the main reason he decided to propose three domains. Now, we notice that fungi, plants, and animals are just little branches on the great tree that is Eukarya. There's a bit of a problem here. There's also other groups that aren't labeled on this diagram in between them that were all counted previously as Protista. Well, Protista is defunct as a result of this, and the number of kingdoms that eukaryotes should be split into is still under debate. Archaea, initially he only identified kingdom Archaea within domain Archaea, and kingdom Eubacteria within domain Eubacteria. This has changed, and it is still under debate to this day. So, for a given contest, you will usually not be asked how many kingdoms are there or how many domains. Well, you can be asked how many domains there are, but how many kingdoms? Well, since that's still under debate, well, yeah, you're not going to be asked that usually. Unless, of course, they're being inconsiderate on purpose, but hey. So, in summary, most biologists can recognize classifying living things using 6 kingdoms and 3 domains. New kingdoms are being drawn up all the time. However, if you talk to somebody about, oh, I have a lot of protists in this sample of water, or I have this many cells of protists per liter in this water sample, they will, for the most part, understand. Because even though Protista as a group is defunct, it is still such a convenient term that biologists, for the most part, let it slide. This is kind of like how the IUPAC, the International Union of Practical and Applied Chemists, or whatever it actually means, so the Chemistry International Organization, pretty much, they accept the terms of water and ammonia. Well, these are terms that are just so used, so traditional, that they are grandfathered in. Because changing them is too much trouble to be worthwhile. Now, if you talk about protists to a biologist, they will understand what you're saying. Though they may ask you what type of protists are you specifically talking about, because say you're talking about a drinking water sample. Saying, oh, there's this many cells per liter doesn't mean anything when it's, say, paramecium versus something that might cause diarrhea. Yeah, I think that's pretty obvious, right? So, The most easily recognized kingdoms are Animalia, Plantae, and Fungi because they're the most obvious to the naked eye, pretty much. Protista is the only former kingdom that does not represent a clade, or to be more specific, not a monophyletic clade. Now this is dismantled now, and it's only used as a catch-all term of convenience. Eubacteria and Archaea are difficult to distinguish by just light microscopy because they are small and they have few, if any, easily visible internal structures, unlike eukaryotes, which are highly compartmentalized within the cell. The Six Kingdom three-domain system was based on phylogenetic relationships revealed by genetic evidence in terms of, hey, Archaea seem to genetically have diverged less from eukaryotes than either one has from bacteria, which means that we need the domains to make the prokaryotes separate from eukaryotes without failing to make the eukaryotes a clade. Early on, before the identification of Archaea versus bacteria, there were 5 kingdoms. Even further before that, we had even fewer kingdoms, namely Protists and Monera, as it was called at the time, were not identified, so we had 3. And at first, we had 2 kingdoms, Animalia and Plantae. So as you can see, we have come a long way in terms of classifying the broad strokes of life. So that's it for this section. And we'll talk a bit more about the mostly from the smallest on up next time.